us. So we're just very excited to have this space and this presence here at Trent and to share Stephanie's research with you. So I'll turn it over to you.
Okay, thank you. It's great to be here. I am a biologist, so I do study probably some things differently than the researchers here or the expertise in the room. But the way I've set up this lecture is to make it consumable for everyone, but let me know if you have questions as I'm going through it. Aging in general, everybody's been curious about it, for a long time. Even exemplified by this picture or this painting from the 1500s where this artist has depicted the Fountain of Youth. And one thing I'll explain to you what's happening in the picture here. So elderly women specifically are going into the Fountain of Youth on the left, where you have all these dark rocky cliffs, and then on the right they emerge out of the Fountain of Youth, very youthful, and on the right there's more, healthy vegetation, lots of food. And one thing that I actually read while kind of learning about this painting was that the men never had to go in the Fountain of Youth because it was just by association of being with women that they would also become youthful. So, aging, anti-aging, you see it everywhere on the news, in commercials. It's just an inherent question that all humans have about why do we age? Can we slow it down? And then in the extreme examples is, can we reverse it? So reversing aging is kind of almost a sci-fi field of biology, but there are researchers that do it. I'm kind of going to approach this more from a kind of slowing aging perspective. So one thing that you have to consider when you do study aging is the population dynamics as a whole. So this graph here is showing you two different pieces of information. So in the white bar graphs, you're looking at just the global population. So no question about it, the number of people on Earth is increasing. But then the blue and the pink lines are showing you the ages of the population. So it's shown as a fold increase versus 1950, but basically the number of people that are 70 and up is, much higher than the number of people under 70. So that's not necessarily a bad thing. I'll get to some potential, implications of this. But another piece of data I wanted to show, also from this same paper, was that the number of countries with these, population demographics are pretty significant. So each dot here is showing you a different country. I don't know if this laser will work on this. No. So each dot here is a different country. And so basically in the 1950s, about 200 countries had a population of 70 plus at about 10% or less. In 2020, this ratio has started to change. Now we see more countries with, between 10 and 20% of their population being 70 plus. And it's predicted in 2100 that over 20%, around 124 different countries will have a population of 70 plus, over 20%. So the number of people that are living into their 70s, 80s, and beyond is increasing. And again, that's not a bad thing, but what is relevant for my research is understanding the diseases that go with it. Maybe this is broken. Oh, there we go. Okay, so here's a general average life expectancy showing, the average population lives to be about 73 in this example. So that's fine, but we know that people don't maintain their health throughout that whole 70 years that they're alive. And so what some biologists in aging want to address is this so-called health gap. Basically, there's a difference between lifespan and health span. Sure, you can live a long time, have a long health span, have a long lifespan, but does that mean that you're also going to have a very long health span? And what we see is that, around your mid-60s is that your health is going to start to decline. So that could mean increases in cardiovascular diseases, increases in musculoskeletal problems like falling, leading to bone breakage, as well as neurocognitive impairments. So from my perspective, I'm not interested in necessarily increasing longevity, but I'm interested in addressing this health span gap. So have any of you seen this picture before? Yeah? Do you remember what it is? Oh yeah, you remember my class, so you would know what it is. Okay.
So if I remember correctly, it's like the amount of UV damage on one side of the face and how it affects how the skin ages.
Yeah.
I don't know, something to do with the sun.
Yeah. So when you look at this man's face, you can see that the left and right sides don't quite look the same. And so this is from an article from 2012 profiling skin damage from the sun on truckers. So basically this man is a trucker, so one side of his face was always exposed to the window, getting lots of sunlight, whereas the other side was not. Another anecdote here is showing a twin study and the effect of smoking. So these are two different people with the same DNA, but one sister, this sister, smoked 17 years longer than the other one. And so what I'm trying to kind of get across with these examples is that it's not just about your DNA that can affect your lifespan or health span, but environment has a major role in regulating that. So this could be environment, affecting specific cells, say cells of the skin, or maybe whole body environment like that of a smoker. So I'm going to tell you about 3 different stories or projects that I've been working on. One is not really cellular, but I think it's a really neat story to tell. It'll be more on epidemiological data. The second is a mouse model of cardiovascular disease. And then the third is probably the most of sci-fi of the 3. So we will actually look at longevity using this tiny water flea called Daphne. So the first one is based on a 4th-year thesis project by one of my students from this past year. And I'll give you a little bit of preamble to the background of why this study kind of took place. So it's focused on Métis citizens, and what was previously reported, and would probably come to no one's surprise, is that cardiovascular disease is much, much more prevalent in many indigenous populations. And so this study focused on just the Métis Nation of Ontario, up to 2012. And what you do in this kind of situation is you collect what's called registry data. So Métis Nation of Ontario, they collect registrants basically to keep track of who is identifying as Métis, and that's very different from census data or like self-identification with the government of Canada. So the Métis Nation of Ontario is kind of like a self-governing body, and so they kind of keep a database of all of those that identify as Métis. And so there's that data, the registry data, and then there's also all of the health data that's collected through your health card through the Institute of Clinical Evaluative Sciences. So anytime you go to the hospital, doctor, they're keeping track of your health through your health card. And so in this study from 2015, what they did was cross-reference data from people that were registered with MNO, and also people that had gone into the hospital with one of 5 different cardiovascular related diseases. And this data here, they compared cardiovascular disease outcomes in 5 parameters. So acute coronary syndrome, congestive heart failure, cerebrovascular disease or stroke, atrial fibrillation, and hypertension. And they looked at not just the Métis identifying citizens, but also the general Ontario population. And basically in all parameters, whether you're looking at prevalence, so total number of cases, or incidence, the rate of new cases, These diseases were all upregulated in Métis citizens. So this is where Sebastian's thesis project comes in. He was interested in doing a kind of survey-based project, but through his own initiative and connections with MNO, it turned into this much bigger project involving the MNO as well as ICES. And so he was able to start digging through newer data, because this only goes up to 2012. So now he's been in the process of analyzing like from 2013 on, basically. So I'll tell you about some of the data that he has found. And for a bit of background, he, for his project, looked at just 2 of the 5 cardiovascular diseases that we could have studied. So one of them is hypertension. And you can think of this as kind of a preamble to worse cardiovascular diseases. So hypertension is narrowing of the blood vessels. That's going to have major implications on heart function, possibility of stroke, or problems with other vital organs, like here showing your kidneys. The other parameter he looked at was heart failure, and in this diagram we're looking at something called hypertrophic heart failure or cardiomyopathy. So in a normal functioning heart, you have a nice ventricle with a normal amount of cardiomyocytes that are an appropriate cell size. But if something were to go wrong, like say you have a heart attack or hypertension, maybe your heart isn't able to circulate blood through your body as well as it should. So it actually adapts and starts to grow in size or go through hypertrophy. And that is good for a little while because it gets more blood circulating through your body, but eventually it's not going to be good. So that can lead to congestive heart failure. So I'll show you a couple tables and then bar graphs about some of the data that he's collected from the years 2012 to 2020. And first I'll go through the hypertension data and then the heart failure data. But basically the way he set up his tables was he looked at the population of Métis identifying or MNO registered citizens with no hypertension or hypertension, and then from there broke it up based on age. So he has 3 bins, 20 to 39, 40 to 64, and 65 and up. And as many other studies have reported, the prevalence of hypertension increases with age. Another thing is that there are more men with hypertension than women. Likewise, when you look at heart failure, you see the same kind of trends where it increases with aging and also is once again more prevalent in men. The thing about heart failure is because it is more severe than hypertension, we just bin based on 40, 64, or 65 and up, because not many people under 40 should have heart failure. If you do, that's like a major problem. So that's why you see there's 3 ages here, but only 2 below. Okay, so we're seeing the same kind of trends as what people had previously reported. What he also did was track these changes over time, so from 2012 to 2020. And right now we're looking at crude rate per 100 citizens, so like 23 out of 100 citizens in 2012 would have had hypertension, but this has increased to like approximately 28 by 2020. So we have hypertension and heart failure more prevalent in aging populations, more prevalent in men, and this is increasing. Another way that we can analyze the data is by breaking it down into age cohorts as well as male and female differences. So this graph is a little bit more complicated, but I'll go through it kind of step by step for you. So this is hypertension. So people 20 to 39, there was no real increase in the number of cases. So that was pretty static. Even with the older cohort, yes, there's more of them, but there was no general change in the numbers of people between 40 and 64 with hypertension across this time point. But the aging population is the one, at least the 65 and up population, is the one that seems to be responsible for this increasing trend. So it could be a few things. Maybe people are actually just living longer, or That could be what's responsible for this increase in the number of Métis with hypertension. The second part of this is the sex differences. So the dashed blue line is men, and the dashed red line is women or females. What this turns into, though, if we actually look at the numbers, like say from 2012 to 2020, is pretty dramatic. So we see an increase of 32 or 36% hypertension for women or men just within that 8-year period. And that sounds pretty high to me. The next thing to look at is hypertension. We see similar trends where that 65 plus cohort is the one responsible for driving this increase in prevalence. The thing about this, though, is if you look at just 2020 versus 2012, that translates into basically doubling for women, so 114% difference for women, or tripling for men almost. 172% increase in heart failure in Métis men just in that 8-year period. So this is definitely some striking data that I'm really, proud of Action for analyzing, and thanks to our partners M&O and ICES. But to summarize, at least for this part, I've just shown you the rates of hypertension and heart failure, how those have increased in Métis citizens in 8 years. One thing I didn't talk about was that he also analyzed socioeconomic status, and that seems to have a bearing as well on the rates of these different cardiovascular diseases. And the way that you can do that actually is by looking at postal codes and kind of cross-referencing income quintiles with that. So cardiovascular disease burden remains high in Métis citizens. There's still 3 other kinds of diseases that remain to be analyzed, as well as comparing that with the population of Ontario as a whole. Okay, so the next part of research that I wanted to share with you is more so about skeletal muscle, which is what I've been studying for a long time now. And in this disease, I'm looking at how problems with your heart lead to problems with your skeletal muscles. And one of the syndromes that can develop is extreme weight loss. Could be because of fat loss, but it can also be because of muscle loss. And if you have a condition like heart failure, they call it cardiac cachexia. There's also like cancer cachexia, which is very prevalent as well. Basically, any kind of chronic inflammatory disease can really reduce your body mass. And we know that once body mass starts to go below a certain threshold, that can really increase your rates of mortality. So it's an important phenomenon to understand. So here we're just looking at a cartoon representation of these changes in muscle mass, either in a healthy person, an older person. Sometimes we call it sarcopenia, which is age-related muscle loss, but in cachexia, it's even more severe. So what some other undergraduate students in my lab wanted to do was look at whether there's any sex-specific effect on cardiac cachexia. And the reason for that is because we published a review article looking at heart failure, different mouse models of heart failure, and whether they looked at male and female mice and what kind of differences they saw. So what the literature told us is that estrogen seems to be cardioprotective as well as anti-inflammatory. So in this diagram, I know there's kind of a lot going on. Basically, estrogen can reduce heart failure, reduce cardiovascular diseases, and we're wondering if by reducing these risks of cardiovascular disease, whether that has any kind of consequence on affecting muscle as well. Another part of this, like I said, is inflammation. So estrogen is thought to be anti-inflammatory. So by dialing down inflammation, maybe that's reducing this muscle wasting phenomenon. And this kind of ties into aging because around that time of menopause, estrogen levels tend to drop. And using mouse models where you overoptimize young mice, reducing estrogen, they actually have worse cardiovascular outcomes than normal female mice. So in the model that we chose, we used something that is kind of like a heart poison, I'll just phrase it that way. And you can inject it somewhat easily into mice using subcutaneous injections. And over a course of 8 weeks, this should lead to, loss of mass, problems with the heart, problems with endurance, etc. So we used this type of protocol because it was easy for undergraduates to do with just like a simple injection. So they monitored body weight over the 8 weeks, and then at the end of the 8 weeks, we started to analyze the mice looking at things like endurance and so on. And what we thought was that based on this idea that females, potentially because of estrogen, seem to be protected from heart failure, we would see this in our model too. So we thought the females would have like normal body weight, whereas the males would have a worse outcome. And so these are the 3 undergraduate students that worked on it, Abby, Alex, and Vivian. And so what they found, thankfully, was some differences. And I'll talk first about this one on the left. So remember, we did these injections for 8 weeks, so we monitored them from time 0 to the end of that 8-week time point and weighed them. And we have male control in black, the treated male group in blue, female control in purple, and female treated in black again. I'll have to change that coloring. But what you can see from here is that males, the treated males, had a much lower body weight, kind of from around weeks 4 and on. And if we just plot raw body weight at the end of the 8 weeks, you can see that there's a significant difference in their overall mass. So they're being injected with the same kind of toxin, but there's a sex-dependent difference in their rate of weight loss. Another thing that we did was test their endurance as a way to kind of measure muscle function. It's not perfect, but it's one way that you can do it. So we use this treadmill on the right. You put the mice into these, separated treadmill kind of cages, and then this treadmill moves and they have to run to keep from falling off onto the back. So we can measure how long it takes them to, tire out essentially. So here we're looking at the distance they ran, male and females, time to fatigue. Basically, if they reach a critical zone that's really close to this falling off point, we call that, the fatigue zone. We can also calculate measurements of work in joules and power in watts. So what we saw here was a little bit different than what we saw with whole body mass. So remember, we saw a body mass decrease in males, but not in females. Here it seems like the drug that we gave them was affecting females more so than males. So at least in these 3 parameters, distance, time to fatigue, and work, there is a sex-dependent effect in females. We saw the opposite trend in power, and I'm trying to figure out what might be the cause of that. When we do calculations for power, it's a combination of how long they ran, like the distance they ran, as well as weight. So power is the only one that takes into account weight, so I think that there's probably going to be some more interesting data to kind of analyze from here to explain this. So that's all I'm going to talk about for that part. So to summarize, we see sex-dependent differences in this heart failure model of monocrotaline or MCT, and it's the first study to ever directly compare the 2 sexes of mice, even though it's a very widely used kind of drug. So this could have some bigger implications in the field of skeletal muscle biology. We're still trying to figure out what's going on with the endurance, but like I said, I think just some more analysis is required. Okay, and then the third part is probably the more technical of the 3. So let me know if you have any questions. But this is a very famous cartoon diagram from, at least in my field, like of aging, from a very famous paper that reviewed all of the sources of aging and put it into these like 3 different causes. So you have what are called primary hallmarks of aging, which could be problems with your DNA, genomic instability, problems with telomeres, which you might have heard of from the news, and epigenetic changes, problems with your proteins or loss of proteostasis. So these are like the ultimate causes of aging, according to this review, and then that can feed into activating these secondary causes and cell problems. So, for example, mitochondrial dysfunction, the powerhouse of the cell, or problems with your stem cells. And so what I'm going to focus on for now is basically nutrient sensing and how that ties into aging. So deregulated nutrient sensing is kind of the secondary parameter here. So in this study, they fed 2 different monkeys, I think they're monkeys, a low or a high calorie diet, like a Mediterranean versus a westernized diet. So they're the same age, 25 and 26, like close enough. But Canto here on the left had about 2,000 calories a day, or well, the equivalent of a human's intake of 2,000, whereas Owen had the equivalent of about 3,000. And so what they saw, maybe you can't see because we don't really look at monkeys all the time, but basically Owen is doing much, much worse in terms of just overall physiology. He's hunched over, his hair is all discolored, so he's not aging as well. So in terms of the molecular pathway that ties into our diet, I'm just going to do a quick overview of some of the things that happen. So a whole bunch of organs help to regulate and sense like the food input in your body and turn that into cellular growth or storage. So we have, input from the hypothalamus in the brain, the endocrine system from the pituitary gland, and then down here we have, say, our target cell. And the pathway I want to talk about here is this IGF-1 pathway at the bottom. So this is a molecule that circulates in your system and is going to eventually bind to a receptor called the IGF-1 receptor and tell that cell whether to grow or not. It activates a very important pathway called the mTOR pathway to do this. And what studies have shown in different model organisms is if you knock out, basically genetically remove the IGF-1 receptor, you're going to increase lifespan. So you can see this in the worm. It's called the worm, but it's not like an earthworm. C. elegans. So if you knock out the equivalent of IGF-1 receptor, DAF2, they live longer. If you knock out the receptor in flies, they live longer, and even in mice, which would be our mammalian equivalent. Another thing within this pathway is this drug called rapamycin. This drug can shut down this whole pathway. So even if you've eaten a meal and your cells are going to be told to grow, rapamycin can say, no, not right now. It's not time to grow. And so rapamycin has kind of become important in longevity studies because it can also improve lifespan. And so this is where our future work may go with respect to Daphnia. So Daphnia are this small water flea, and people have started to use it as a model organism for longevity. And what's great about it is Paul works on Daphnia and in nutrient stress actually. And so I'm interested in the aging side, he's interested in the nutrient side. So we're going to try and see what we can do together. But basically in this study, they actually treated Daphnia with rapamycin. So it's a drug that shuts down the IGF-1 pathway. And they did it at 3 different doses. So at a super high dose, you can see that size is different. Whereas at the less intense dose, it's not so much different. And then when they plotted longevity, they saw that pretty much any dosage of rapamycin increased lifespan of these model organisms. So you might have heard of rapamycin through the news, maybe not yet, but now if you come across a news article, you'll say, oh, rapamycin, that sounds familiar. These are just some of the recent articles that I pulled. It's a very popular drug for anti-aging studies. I'm not prescribing it, don't take it, but it's cropping up in a lot of different studies, particularly because it can limit growth. So that means it can also potentially limit cancer growth too. So to summarize this piece, I guess what I was trying to get across was there are studies that show if you eat less, you stay smaller, but you live longer. Whereas there are studies out there that say, you know, keep your normal diet, but just take this drug rapamycin and you'll get the same effects. So those were the 3 studies I wanted to go over for you today, kind of going from whole human to mouse work to longevity research. And maybe what you can also take away from this is that aging is going to be influenced by, you know, your diet, also your exercise, maybe even your sex. And so you have to eat right, you have to take care of your body. But I saw a talk by someone else that does aging research, and she said, like, she would interview centenarians, so super old people, and some of them would say, like, they smoke a lot, they drink a lot, and yet they still live to be like 100. But one of the main things that they talked about was having good familial connections, like having a good society, or, you know, good people that they can spend their time with. That speaker actually highlighted how much that shone through during the COVID pandemic, when those ties were kind of severed, and how that can just, you know, increase mortality, basically. So it's a whole picture kind of thing, and I think that TCAS is doing a good job of incorporating all of these different sides of aging. And so with that, I'll say thank you to all of my different people, my different collaborators. One project I didn't get to talk about was on muscle stem cells, but that's another thing we have going on. And yes, thank you.